So I'm going to talk just about very quickly about my own journey here at the University of Chicago, but also overview of the Cancer Center, and then really focus today on the mechanisms by which cancer is initiated in a cell, and then goes on to full-fledged metastatic cancer. So I've been at the University of Chicago since 1989. I had developed a drug while doing my postdoc with Tony Pegg at Penn State, and we were bringing that into clinical trials. And so I was hired into the Department of Medicine at the University of Chicago, and I had worked on this DNA repair protein that we were targeting, that we had developed a drug against, that helped to create an environment in which we reduce the resistance. So brain tumor chemotherapy was far more effective if you use this drug ahead of time. And that went into clinical trials. And I worked on that DNA repair protein for about 15 years. And around 1999, the technology had evolved in such a way that allowed us to look at more than just single proteins, single genes, single RNA, and it allowed us to look at many different changes at the same time in a cancer cell. And so we transitioned into this field of pharmacogenomics that was just emerging then because of this new technology. And at the University of Chicago at the time, we had, and we still do, have extremely strong genomics department. And we also had really strong clinical trialists. We had a phase 1, phase 2 clinical trials going on. And then we had a very strong pharmacologist, and all of us came together. We applied for a grant that was $2 million a year, and these were 5-year awards, and we ended up getting that grant. We were one of the top grants, and we ended up getting it that year. We got it renewed a second time. So we had 15 years of funding in this field of pharmacogenomics, and it was extremely exciting because of being able to use this new technology, but also because our goal was to identify how genomics or DNA plays a role in making an individual highly susceptible to the toxicities associated with chemotherapy and radiation, but also understanding how our DNA, our germline DNA, makes us more likely to respond or not respond to chemotherapy. And so my role in all of this was to build cell-based models. And one of the models that we built that was really useful was taking blood from patients who had severe toxicities to chemotherapy and reprogramming them into induced pluripotent stem cells that then allow you to genetically engineer and create many different cell types with different changes in the DNA to study them very specifically. And so that's what I've done. But at the same time that I was doing all of this, I've always been interested in education. That's why I'm in academics. And so in 1990, I had a young girl, Terza Mitchell, asked me if she could work in my lab. She was in high school, and she ended up winning the city and state science fair. And it was a lot of fun for her to be in the laboratory. She's since become a nurse and works in IT. But because that was fun for me, I then served for many years as a volunteer at the American Cancer Society. And we built a program from originally 4 students up to 30 students. We put students throughout the state of Illinois in different universities. And that program, because of funding, they no longer are doing that program. They stopped that about 7 or 8 years ago. And the University of Chicago has created high school programs. We put students in universities all throughout the state of Illinois, but we also have other programs for students that are from under-resourced areas called Chicago Eyes on Cancer and She in Oncology. And these students, for Chicago Eyes, for example, we take high school students, undergraduates, and high school teachers from the surrounding area. And they come for 2 summers and work for 8 weeks each summer. And then we do activities throughout the school year. And it's really great because we help build capacity for our neighbors in Chicago South Side. So the University of Chicago Comprehensive Cancer Center is one of only 2 National Cancer Institute comprehensive cancer centers. It's funded by a grant by the NCI, and it's our mission is to develop innovative and collaborative research to discover the determinants of cancer, to develop cures for cancer, and to prevent cancer. And we have over 200 members that are part of the Cancer Center. I was a program leader for 20 years. I'm the Associate Director for Education for the past 7 years. We have 4 different programs that span different areas of cancer which bring together groups of individuals that are sort of like-minded in their research efforts, that can work together and provide, seminars and expertise to one another. And these are just the 4 different programs that we have within the Cancer Center. We also have within our Cancer Center about 13 different shared resources, which are core facilities that provide us with resources and expertise to help with our research. So we each can't buy our own next generation sequencing machine. So we have a number of them in our genomics facilities. We have individuals with expertise in statistics to help us with our biostatistics and in the mouse facility, as well as immunology and so forth. So all of these different core facilities that are supported through our National Cancer Institute grant are utilized by members within the Cancer Center to help provide, research progress. So cancer is both common and deadly. There's approximately 16.9 million cancer survivors in the United States, and by January 2030, we expect there to be about 22.1 million. In 2020, there were 1.8 million new cancer cases and 600,000 cancer deaths. And approximately 1 in 3 women and 1 in 2 men will develop cancer in their lifetime. Prostate is the most common cancer in males and breast in females. Lung comes second for both males and females. And then colorectal cancer is also quite common in terms of the incidence of cancer. You can see that leukemia is not as common. It's only about 3 or 4% of all cancer cases in 2020. In addition, this is showing the mortality of cancer, and that varies considerably across different tumor types. So the 2020 estimates show us that lung cancer still is the most deadly cancer, both for both men and for women, with prostate and breast respectively in second place for the percentage of cancers that cause, or this percentage of individuals that die of cancer. So at the University of Chicago, we have individuals that work in the entire cancer continuum, from prevention to screening, diagnosis, treatment, recovery, survivorship, as well as end-of-life care. And you can imagine there's also individuals working on psychosocial and palliative care throughout, from diagnosis to end-of-life care. So what is cancer? Well, the American Cancer Society identifies it as a series of genetic mutations in cells that allows them to grow out of control and invade other tissues. It's really a collection of diseases. So it's about 100, we consider it to be 100 different diseases. And sometimes we've talked for many years about no longer calling it breast cancer or colorectal cancer or prostate cancer, but calling it by its molecular profile instead, P53, and there are many different changes that occur within the cancer, mutations that I'll talk about in a minute. But what happens is this collection of diseases arise when the processes that control normal cell growth and division and lifespan go awry. So as a result, the cells are starting to multiply uncontrollably. They fail to die when they should die. They mobilize other cells and tissues, such as blood vessels and immune cells and other types of normal cells, and give the tumor an advantage to continue to grow in an environment where it should be dying off or staying stationary. So the mutations that are occurring in DNA are obviously then causing it to, allowing it to do things that it wouldn't do in a normal environment. And as the cancer grows and progresses, the cancer acquires specific changes that along the way that allow it to do even more damage and allow it to grow at other sites. So this is just a liver cancer, and you could see these are macro metastatic lesions. So this is, the tumor is quite large throughout the tissue, and it starts to take over the normal liver function or the normal function of the tissue. And so the process that occurs is that there are originally, we get a mutation within our DNA from the environment or from normal cell division. These is just an example of normal cells growing on a basement membrane, and they're very happy and they're all uniform shape. And you see the nuclei, which is shown here in kind of like a yellowish white with the DNA inside it, and everything's good here. But now you start to get some dysplasia because a mutation occurred in one of those cells, and the dysplasia allows more cells to grow in what should have been the space for just these 4 cells. And then additional mutations occur where now you have a carcinoma in situ, and you can see no longer are the cells have the same uniform shape. All of a sudden there's different shapes, and it's not well depicted here, but what often happens is the DNA is replicating so rapidly within the nucleus that the nucleus becomes larger and larger, and the cytoplasm, which surrounds the nucleus, becomes smaller and smaller. So you have these non-uniform shape, you have the cells growing uncontrollably. And then at some point you have a situation where mutations occur and allow that cell to leave its normal organ. That would never happen with a normal cell. It leaves it and gets into the lymphatic system. And now it's able to survive in this lymphatic or the blood vessel and transverse to another tissue. And this can then create what we call micrometastases, which is a cells growing in another organ somewhere else in the environment, which can eventually also cause an additional tumor. So I often have a situation where, somebody will say, my mother had breast cancer and now she has brain cancer. And that's probably not what happened. She probably has breast cancer that has now metastasized to the brain. So how does DNA damage occur, and it can lead to protein malfunction? Well, DNA is found in every cell. There's 3.1 billion base pairs, and these are the instructions that make all of us who we are. So the reason I have green eyes is because of my DNA, and we are all 99.9% identical in terms of the sequence of the DNA. And that 0.1% difference makes all of us different in how we look, how we act, in oftentimes in our personality, and other traits that we carry with us. And there are small segments of this 3.1 billion base pairs which create the genes, and the genes are sort of then go into what are called RNA or messages, and then create proteins. So I often think of this sort of like as the CEO of the company is making all the decisions, that's the DNA, and creating sort of the strategic plan. The RNA is sort of all the managers who are providing that message to all of the workers who are really doing all the work. But what can happen is when you have a change in the DNA and things go awry, or a change that occurs in a critical DNA component that's important for cell division, it will affect what the protein looks like or how the protein acts. And it can cause greater cell growth or change in cell structure and cell death, so that it no longer dies, because that protein that was critical for cell death is now altered in such a way that doesn't allow it anymore to die. So these are all really, really important components of regular normal cell division, but we can have mutations in DNA that alter the way in which these occur. So what causes the mutations in DNA? They can be chemical insults from the environment. So UV radiation, we can have toxins from carcinogenic compounds, maybe additives in our food, from X-rays, from viruses. We know that human papillomavirus causes cervical cancer. But we also have our DNA is always replicating. That's what normally happens, as we live every day, and our cells divide and get replaced. And when you have replication occurring all the time, sometimes there's mistakes that are made. You could have DNA repair mechanisms that are supposed to find those mistakes, maybe make a mistake and not identify a particular mistake that occurred. You could have random mutations occur in our somatic cells. As we age, mutations just occur in normal aging. And there's also other mutations that can be passed through the germline. So 10% of all cancers are due to a DNA change that came from our mother or father. So examples of this might be BRCA1. So if you actually have BRCA1 mutation in your DNA, you have a 50-50 chance of passing that on to your child. If they carry it, they have an 80% chance of getting breast cancer sometime in their lifetime. So these are called inherited mutations. And it's also found in certain types of colorectal cancer. And they increase the likelihood of you getting cancer because you already are born with certain mutations. And so these are just the damages throughout our environment. Obviously, cigarettes, I didn't even mention, are highly carcinogenic and can cause DNA damage. So I mentioned that the human genome is 3.3 billion base pairs. So I often think of this sort of like, if you made it linear, it'd be very, very long, but the DNA is crumpled up in three-dimensional structure in each one of your cells. And it's the same in every cell in your body. But if the message gets messed up, so let's just say this is the normal message, the big red dog ran out, you could have a single mutation. This is the most common. This is very easy to create a single mutation because it's just a small change in the DNA sequence. And so you still get the same message, you kind of can get it, but it's something is wrong, it's off. You can also have splice variants where a part of the message is removed of the DNA. You can also have what we call deletion. So even a larger part of a message is cut off or taken out. And then you can have insertions where something is put in, but all of these cause the message to be different than the normal message. And then we have what's called copy number variation, where you could have copies of the same gene that occur over and over, and you get this overexpression of a gene. So if that gene was important for cell growth, and now you have lots of it occurring in your cell, then you could be, have this cell growing out of control. So chromosomes, which are thread-like structures within the nucleus, where your DNA is compacted into many different chromosomes, well, 22, and then you have X and Y to determine whether you're male or female. These are the DNA with protein around it. And what you can also have is a large chromosomal change in the DNA. So these are called structural changes. And one great example of that is a translocation or a gene duplication that I just mentioned. So a translocation is a part of one part of the DNA. And these are like large segments of DNA that actually get moved from one chromosome to another. Or you could have gene duplication where you have a gene here, but then it gets duplicated multiple times. So this can occur anywhere in your DNA. And one great example is Janet Rawley's work at the University of Chicago, where she worked in looking at these chromosomes in leukemic patients. And she one day walked down the hall and saw Harvey Gollum, and she said, I think I've identified that there's this change that occur on chromosome 9 and 22. It moves over from 9 to 22. And so she really transformed the view of cancer because she was the first one to show that there is evidence that cancer is a disease that's caused by alterations in DNA. Prior to that, there was so much work on viruses and so much work on other areas that we thought that these external sort of viruses or bacteria or something was changing and causing cancer. And so this really sort of was the first time where we knew it was within our own DNA. And so this change that she showed, which is chromosome 9 to 22, produced what they called the Philadelphia chromosome. And the gene BCR became very close to the gene ABL. Normally they'd be on two different chromosomes and be completely independent genes. Now that they're right next to each other, they created a brand new gene called BCR-ABL. BCR-ABL caused leukemia. that or caused chronic myelogenous leukemia. So in 1972, she was providing the first evidence that cancer is a disease caused by alterations in DNA, and really opened up an entire field. So this Philadelphia chromosome, she was looking at it like she used to describe it sort of as cutouts, like doll cutouts, where she was looking at all of these different things, these chromosomes, and evaluating what was different in the leukemia cells versus the normal cells. So BCR-ABL, once that was produced, the actual protein from this new gene that had not been there before, and it was only present in chronic myelogenous leukemic cells, they put it into cells and they put it into animal models and saw that it produced cancer. And once you have something really, really unique in the tumor cells that's not found in any of the other normal cells, you can create a targeted therapy. So there was a development of the drug Gleevec that inhibits BCR-ABL, and that was tested in clinical trials. And Gleevec became really the standard treatment for chronic myelogenous leukemia. It was extremely effective because no other cells in the body have it. It's just these CML cells. And since Gleevec, there's been, of course, other drugs that have been also developed, not only against BCR-ABL, but against other mutations that occur when this is resistant, and other targeted agents. So the specific genes that regulate the properties of cancer are ones in which they code for proteins that promote cell growth or division in normal cells. So when certain genes, when these oncogenes, we call them, are mutated, the growth signals are active and cannot be turned off. It's sort of like, a car which doesn't have brakes. And then we also have tumor suppressor genes where we code for proteins that either prevent cell division or allow programmed cell death. And when these are mutated, the growth inhibitory signals are no longer active. So in either case, whether it's an oncogene or a tumor suppressor gene, in either one of these scenarios, you have what we call sort of uncontrolled cell death, or uncontrolled cell division, or abnormal cell division. So that occurs in the very initial stages of creating a tumor cell or a cancerous cell, which allows a cell autonomy. It's no longer listening to its neighbors. It's no longer following the rules of the environment that it's in. It has self-renewal. It evades death. It evades immunity. But then it moves on and becomes even more dangerous because it now has mutations that allow it for detachment. It allows motility, invasion. And a tumor cannot do anything once it's the size of a pea, it has to have a blood supply in order for it to grow, continue to grow, and to spread. And so what it does is recruits its own blood supply from surrounding blood vessels. And so that now the blood supply is within the tumor and allows this tumor cells to go into the blood supply or the lymphatic system and travel to other distant sites. And it evades the immune system, which, is present to protect us from exogenous or environmental toxins. And the one thing that's really interesting about cancer metastases is that each tumor metastasizes to very specific sites. It just doesn't metastasize anywhere. It has to find a fertile ground somewhere else. So, for example, breast cancer only metastasizes to the bone, to liver, to lung, or to the brain. It doesn't go to the colon, it doesn't go to the pancreas, it doesn't go to the ovary. It literally only goes to those sites because those sites provide a fertile ground for breast cancer cells to grow and continue to create tumors within that site. Brain tumors never metastasize to other sites. People die of brain tumors, but it's never in any other site in their body. Ovarian cancer goes to the pleural region and causes liver metastases, but it doesn't go to other areas like the bone or the lung or other parts of the body. And so cancer research And, you know, all of you have been involved in providing support for individuals to develop new targeted therapy. You know, for decades, we've been using chemotherapy that was developed in the 50s and 60s and 70s. And at that time, our knowledge of cancer was just that, well, these cells are growing and replicating out of control. So let's just find drugs that target cells that are growing fast. And that's what many of those chemotherapy, paclitaxel, cisplatin is the most common, is part of platinum agents. These are the most common group of drugs that are used worldwide for cancer. And they were developed just in laboratories against tumor cells that were growing very rapidly. But now many, many new agents, which many of you have through the CRF have funded, have been developed. And that is really to receptors that are unique to hematologic malignancies or leukemias and lymphomas, but also to solid tumors. So a great example is HER2/neu, which was found on breast cancer cells and some other cells, and Herceptin was developed, and that has really changed the landscape of breast cancer for individuals that have HER2/neu positive breast cancer cells. So they target specific molecules that are expressed only in the tumor cells. They preferentially kill cancer cells, and they're effective for many cancers, but some will develop resistance because the cancer is really, really smart in terms of finding ways to get around chemotherapy or radiation by creating additional mutations in order to continue to grow in that environment. Oh, I'm sorry. I don't know what happened there. I apologize. Okay, so just to sort of, end here, I have wanted to tell you about cancer genomics is transforming our view of cancer. It was Janet Raleigh who brought us into this field, but the Cancer Genome Atlas is one in which they're looking at 33 different tumor types. That includes 10 rare cancers. They have 11,000 patients. They have 7 different data types, so they're sequencing all of the protein coding regions on DNA. They're doing whole genome DNA sequencing, so looking at introns and exons and gene expression profiling. So they're looking at the message, the messenger RNA. So they're not only looking at the CEO and every single aspect of every decision, they're looking at all the messages that are sent out, and they're looking at the microRNA environment, the epigenetics, and the protein. So all of these different things are being evaluated at exactly the same time. So we know, what DNA changes occur and how that impacts the mRNA, how it impacts the protein, how it impacts other changes within the cell to create the, eventually cancer. And one of the things that we've obviously identified over the last decade or more is that cancer is made up of many different mutations. So it's very hard to target cancer because of the fact that there are multiple mutations. One of the most exciting components that that technology has allowed us to do is look at millions of changes within the DNA at any given time. So this just shows you breast cancer, and this is likely thousands of patients. And what it shows, we call it Manhattan plot because it looks as though it's buildings here, but the peaks are just these are all points that have a very significant p-value. Everything past this dotted line is highly significant. And the x-axis tells you the location of it. So it tells you that on, for example, chromosome 19, there are many, many mutations that occur in breast cancer patients. And these are all present, as well as those on 12, which is really a great example of why it's so hard to treat these cancers, is that, you could target this particular mutation, but then you have other things that are also present. And then one of the things that we've also learned over the last decade that's really, really important when we look at these millions of changes that occur at the same time is that DNA is not linear. We always sort of thought, oh, there's these intronic regions. We called it sort of dark matter. They're not producing proteins. They must not be important. But in fact, they turned out to be really, really important because in these intronic or dark regions that we thought were just, sequences of DNA that didn't matter, they actually contain many of the regulatory elements. And so because DNA is three-dimensional, there could be changes on chromosome 3 that affect chromosome 12. Or on 6 that affect 21. So this is just an example of pancreatic cancer and all the different mutations, and then how they affect other parts of the DNA. So I'm just going to end with a case report, and this is just to give you an example of the the difficulty in treating patients based on population averages, as opposed to looking at mutations, which is the newer and better approach. So this was an individual that came to the University of Chicago who was a 48-year-old African-American. You could see a large breast mass, which is a dominant breast mass here, and large lymph nodes. She had skin thickening as well, and she was diagnosed with triple-negative breast cancer. She had estrogen receptor, progesterone receptor, and HER2/neu were not positive, which meant she could not be treated with any kind of anti-estrogen or hormonal therapy, nor could she be treated with Herceptin because she did not carry those receptors. So the options for treatment can be surgery, radiation, chemotherapy, immunotherapy, and only recently has immunotherapy come into the forefront for the treatment of triple-negative breast cancer. And so this particular patient was treated with docetaxel, paclitaxel, and that's because on population averages, most patients, not all, but most respond to docetaxel, paclitaxel. There were currently, at the time, this is several years ago, there were no standard treatments for triple-negative breast cancer. So this was a reasonable approach. But you can see now we're looking down at the tumor here in the left breast. Paclitaxel has a lot of side effects. There's bone marrow suppression, there's neuropathy, which is a numbness or tingling and the burning pain in the hands and feet. They can have joint or muscle pain, nausea, vomiting, mouth sores, hair loss, hypersensitivity to the when they receive the drug. They also, which is less common, they could have liver function abnormalities that occur. And in her particular case, she underwent 4 cycles of paclitaxel. There was no change at all in the tumor after those 4 cycles. And we know that paclitaxel is metabolized by the liver, and this woman experienced hepatic or liver toxicity. So when they get liver toxicity, now you have to wait. You can't treat them with anything for several months until that resolves itself. And then, so there was a discussion about, should she be dose modified? Well, she's not responding at all to these, so that doesn't make sense. Should they change the anti-cancer agent? Should they give her a different type of treatment? And so in this particular case, they changed the anti-cancer agent, and they went to adriamycin cytoxan, another therapy for regimen for triple-negative breast cancer. But again, you can see how this is sort of a guessing game. We don't know. We tried this, now we'll try this. And she did get a beautiful reduction in her tumor. As you can see here, the left breast mass is now gone. And then, but I just wanted to illustrate this because so many patients do this, where they go through rounds of chemotherapy, they then get tested and they have no response, and it's exhausting for them. And the question is, what if we're able to minimize the chances of this, exhausting cycle? And the way to do that is through these genetic tests. So instead of just bringing them in and making a guess, we now do genomics. We do both germline genomics, we do somatic mutations or tumor mutations. And the goal is to identify genetic variants associated with high risk for non-response or toxicity to modify the treatment accordingly. So precision oncology is sort of the way that the University of Chicago is now treating patients. The idea is try to get all the patients into this group where the drug is not toxic but beneficial, as opposed to being toxic and beneficial, or not toxic and not beneficial, or the drug is toxic and not beneficial. So the idea is to try to get all the patients through genetic information into this group of having less toxicity and higher benefit. And because all of the work in genetics, or the vast majority, has been done in individuals of genetically European descent, we have a lot of information, and some of that can be translated, but not all of it, into other ethnic groups and other races. So we need to do more work in the area of Blacks and Hispanics and Asian Pacific Islanders, because there's an unequal burden of cancer in the African American community. There are more deaths relative to the number of new cases when you compare Blacks to whites. But the good news is that cancer death rate is decreasing, and you can see that here. This is African Americans have had the most dramatic decrease in cancer death rates. This is shown for whites in the green, and then the red is the American Indians, Asian Pacific Islanders, and Hispanics are in these two blue. So that is good news, and I'll end there by saying thank you. I also want to thank the CRF for the fact that they've put so much money into cancer research, over the past many years, and made incredible difference in what's been able to be done in terms of targeted therapies. And all of the things I've talked about really come from the discoveries that are made from the funding that you support. So I'll stop there and happy to take questions.
Great, thank you. Any questions?
Eileen, can you talk a little bit about the idea of data mining backwards as we talk about separating genomic patient groups? Is there some hope of being able to use some of the data we have to even better distinguish?
Yeah, there's an enormous amount of data that's available. And in fact, with these summer programs that we have, since they're remote, we're actually teaching all of the students to do, the high school and college kids to do R programming and Python, because even if eventually they go into a wet lab and do research in a laboratory with animals or cell lines, I mean, this R programming and the data mining is so critical for our understanding. It's the first time ever we know about millions of changes that occur in the DNA and how that affects the message and how that affects the protein. So, we're working hard to continue to bring individuals into this area of data mining and data analysis. And it's not easy because you use up, like, every day I'm getting an email about I already used up all my terabytes of space, and I'm like, oh, it's so much data that you're looking at the same time. But it allows you to see things in a way that we were not able to before. When I did research on one DNA repair protein, I was very biased. I feel like this is the most important protein and I'm going to study it, and now I'm completely agnostic. I'm saying, when I ask these questions, like you pointed out, looking in a way at lots of different changes, you're unbiased. You're saying, I'm going to let the data tell me what's important, as opposed to me just looking at one particular component that I think is important.
In your case study, you talk about going from one drug to another. How in the process do you, when and how do you contemplate surgery or immunotherapy for a breast cancer patient like this? Yeah,
that's a really good question. Oftentimes if it's metastasized and it's in the lymph nodes or in another organ, surgery is no longer an option, because they feel as though that is not going to cure the patient. You really have to, it's already sort of, they often say the horse is out of the barn or whatever, but it's very hard to cure cancer if it's now metastasized to other organs through surgery. So, you have to bring in radiation, you have to bring in oftentimes chemotherapy, and immune therapy, which is really the last couple of years has completely changed the landscape of cancer. I mean, lung cancer and melanoma, which were deadly, are now, people are living years because of this new immunological therapy. So oftentimes physicians will make judgment calls based on the fact that maybe a person has certain mutations. So if they carry a particular mutation within their lung cancer, then we know that this checkpoint inhibitor will be effective, or we know this immune therapy will be effective. So decisions are really now being made based on mutational profile of the tumor, and deciding based on that. Or, you know, and there's obviously standard of care for certain cancers as well.
Go ahead, Zana.
Okay. Eileen, I've never actually seen that graphic of different sites of metastasis for different cancers. Is there some hope that someday we'll actually be able to create a block for, you know, where it's going as opposed to the site the cancer is?
Yeah, that's a really good point. So, you know, right before that, you know, there's this angiogenesis too. So there were a lot of angiogenesis where the tumor now gets big and, well, I was saying the size of a pea, it now needs blood vessels, right? Now there's a lot of agents that were developed to try to prevent the blood vessels, because now you're preventing it from getting to a metastatic site. It's very hard because once it's metastasized, that tumor that has now so many additional mutations that it is allowing it to grow in a completely different environment. That is, for example, a breast cell, which is now growing in a lung. You know, that just wouldn't happen unless it was completely changed in a way. So it's much more difficult. But there are, you know, there are sort of this attempt through immune therapy to try to get at these metastatic sites. Because really that's, you know, people die because of metastatic cancer, not because they have a primary breast cancer that is only in the breast. It's because it's now in the liver and taking over major organs that are, you know, critical for life. But I think the immune system and these checkpoint inhibitors that are newly developed are really going to make a difference in terms of metastatic cancer.
Is the proliferation of cancers into the body cavity considered metastatic movement, or is it just?
It's called intravasation, where it gets into the blood vessel and the lymph node.
Mhm.
So sometimes it's just in the lymph node and not somewhere else. And sometimes you don't even know because it's single cells that are traveling now from a lymph node to another site. So they call that like micrometastases, where you can't see it under the microscope, but we know that there's likely sites that are present or cells that are present, eventually form tumors and you can see those. But that's what makes it really difficult, is that imaging, you know, you can't image what might be single cells. I mean, it's not till, you know, you get a million cells there that you can actually be able to see it.
You mentioned external influences on causing cancer, and you mentioned smoking, for example, and yet lung cancer still is a big killer. Are there other efforts to determine whether the environment or habits or lifestyles really contribute not only to lung cancer, but other forms of cancer? Or is this simply a chemical phenomenon that occurs without any external influences?
No, I mean, you know, there's a lot of work now, not just in identifying chemicals, but how structural racism has contributed to the disparities that we see in cancer. You know, why is it that Blacks are more likely to get triple negative breast cancer? Why is it they're more likely to die of certain cancers that seem to be equal, you know, in terms of whites and Blacks? So, you know, there's lots of determinants. There's work going on at the University of Chicago evaluating how stress in an environment where you're living, you know, in a high stress environment, how that contributes to cancer. So, you know, there's a lot of variables that are not just chemical. They could be social. They could be economic. They could be, you know, there could be genetic differences just because we're all different in terms of our own DNA, and populations are different in terms of their DNA. So it's many different variables that contribute to cancer and that contribute to the you know, how deadly it is or how aggressive it is, that involve both chemicals as well as the environment as well. But that's a really good question, because that's been a really interesting field to look at the socioeconomic component as well.
Thank you.
is the University of Chicago doing more in those health disparities and looking at that? Can you say a little about that?
Yes, yeah, I'd love to talk more about that. We have, first of all, we have a program called Cancer Health Disparities program through the Cancer Center that just was funded by Komen. So we fund graduate students that work in cancer health disparities. I teach a course with Dr. Olapati every year on cancer health disparities. And I have to tell you, it's very difficult because there's so many students, undergrads, it's a graduate level course, but I always get 30 undergrads that want to be part of that class, and I allow them in, and it's a lot of work, but after that class, because most of them are pre-med, and it's their first exposure to physicians talking about health disparities. And it's their first, they don't even, like when you're 19, you don't even know what health insurance is. You don't even, like you're on your parent, you don't even understand that people don't have health insurance. So this really opens their eyes, and so many of the undergrads end up like afterwards sending me an email. Oh my gosh, I'm now going to dedicate my career. I'm going into, public health, or I'm going into medical school, and this is what I'm going to do as a result. So there's a tremendous amount of work, not only local health disparities, but global through Dr. Olapati's efforts for global oncology.
If I might just add to that too, I can just say kind of anecdotally from my work with different faculty across the disease spectrum at University of Chicago, given our patient population, we have many clinical trials and many different efforts woven throughout all of our programs that really do involve a diverse mix of patients. And there are specific studies looking at disparities. Sue Cohn has done work in that in neuroblastoma. We have some of that being done in prostate cancer. And then of course, there's Habib Hassan's work with the COMPASS trial. So lots and lots of things really across the enterprise. And we could certainly share some more of that if that's interesting to any of you.
I think it would be really fun for you to have a meeting between Blaise Polite and maybe Fumi Olapati, who will provide an argument. They will argue with each other about of what, like the genetics, what are the genetic biological components that contribute to health disparities? And then Blaise will say, this is all policy, it's policy that's impacting it. And he is phenomenal. We have him talk to our graduate students. He's just fantastic in understanding and studying how policy impacts cancer disparities.
Great. Any other questions?
Yeah, not to get too specific, but with respect to factors of the environment, do you have any thoughts on the increased rise in colorectal cancer in folks, under 45 and even under 40? I keep reading more and more articles how this seems to have a greatly increased case counts in younger folks who we weren't really seeing that. I mean, do we think it's what we're eating, or any thoughts?
Yeah, I mean, colorectal cancer is very common in the United States because we have a very high fat diet, and and lots of fast food. Whereas if you look at that particular statistic in Japan or in an Asian country, colorectal cancer is very, very low because they eat a much different diet than we do. So a lot of it is attributed to our environment and the kinds of food that we eat, and also the fact that we have a country that is very much into fast food, and oftentimes that is equated to high fat.
I was fascinated with that one chart that plotted metastasis, that certain cancers only go certain places.
Mm-hmm.
I've never heard that expressed that way before. Is there an explanation as to why that happens that way? Or
well, yeah, and it's interesting. I think I mentioned when I was talking that, you always get somebody saying, oh, my mother had this and now she has this, but really, it's metastatic lesions. And it's because the kind of tumor cell that is initiated in a primary site can only grow on certain ground, and if the ground is a liver cell, it's allowing a certain tumor type, original tumor type, to grow there. But there's a lot of work in trying to understand the extracellular matrix and the you know, which is really the ground or the environment for that to continue to grow. So it has more to do with the actual metastatic site. So the organ in which the tumor has come to, it has to do with that contributes to allowing, you know, the growth of that particular type of cancer. So both things are important. It's not just like the fact that this is a breast cancer. It's that the liver is now allowing us the right soil for the growth.
Thank you.
You said there are hundreds of cancers. Is that, do we
Yeah,
know
I mean,
the specific number?
I don't know the specific number. We always say there's hundreds because, you know, with lung cancer, you have all different subtypes. And even so, you know, before we might have subtyped it one way. And now that we know all the genetics, we're subtyping it a little bit further out. So, you know, in breast cancer, you could have, you know, 6 or 7 different types of breast cancers, or, you know, so all these, at each different organ specific cancer, you can have 7 or 8 different kinds of subtypes of cancer, depending on that molecular characteristics that make up that particular, you know, cancer, lung cancer, whatever it is.
And are we discovering more? Does that go up?
You know,
Yeah,
because we're discovering that maybe only 3% of all breast cancer patients have this particular mutation. Should we make that a subtype or not? You know what I mean? But we could target that. So same with lung cancer. If they carry the ALK mutation, that we can target with this. So it's 100 different diseases if you sort of consider not the organ of sight, but you're considering the molecular characteristics or the genetic profile of it. And that could be increasing with time as we learn more.
Okay.
But the good news is that, you know, each time we get like, oh, this particular mutation is really, really important, then that provides individuals that are doing drug discovery to find ways to target that particular mutation or change. But again, it might only be in 3% of all lung cancer patients that have this particular mutation.
And then Eileen, they do find that mutation happens in multiple cancers, right? Like you mentioned ALK.
Right.
So ALK is in like what's like lung cancer. Again, I think some pediatric cancers, like they really have, once they discover it in one cancer, they realize that that mutation is across other disease types and can apply those therapeutics to the different cancers as well.
Right. Which is why I said at the beginning, there's this, you know, thought that we shouldn't call it breast cancer, lung cancer, pancreatic cancer, that we should now call it ALK positive, blah, blah, blah cancer, or MET positive, or P53, or, you know, whatever the mutation is, because really that provides us with more information about the appropriate agent to use as opposed to the site at which it starts.
Do you have, once I know I'm getting specific here, but do you have any experience with, in my dad's case, he had I think it was an ALK specific lung cancer, and he responded amazingly to Keytruda, and took it for 3.5 years, to the extent that the cancer is 0 activity. They say it's dormant, but he's now gone where he can no longer tolerate the Keytruda. So he's off, he had to stop taking it. Since the cancer is dormant, do you generally find that it will come back, or is it as if it was actually, you know, in a true state of remission when you have to go off an immunotherapy?
Yeah, that's a good question. We don't know. I mean, tumors can be dormant for 20, 30 years, you know, and we often don't know what makes it come back. What, you know, I also have a good friend who had breast cancer in like 1990, and now she came back with lymph node. The same breast cancer is now back, and it's at least 40 years later. How is that possible? You know, I don't know. They stay dormant. What makes it sort of wake up?
But that's still good. I mean, in my case, he's older, so that's a good answer, that the dormancy could be a while, which is.
You know, and so all cancers grow at different rates. So prostate cancer, sometimes they don't even treat it in older men because they're like, you're likely to die of something else because it's such a slow growing cancer.
Doctor, could I ask you a very basic question? So tumors for us lay people sometimes are categorized as malignant or benign. Is a benign lesion, a benign tumor, is the cell malformation such that it will not invade?
So, right, that's exactly right.
Why has it grown?
Yeah, so benign means that it would probably have some mutations that allowed it to grow in a certain site, but it remains where it is. So it's not dangerous in the sense that it's not going to move somewhere else. Whereas a malignant tumor is one in which it can get into a blood or lymphatic system and move to another site.
So malignancy requires for it to be metastatic?
Yes, but you could, but we know, for example, like if somebody had breast cancer only in the breast, if it's an adenocarcinoma, we know it's malignant based on the characteristics. So maybe it didn't metastasize yet, maybe it's not there yet, but they know from the characteristics of the tumor that it's definitely a malignant tumor. Yes, they would know that.
Okay,
Yeah.
thank you.
Okay, any other final questions? It's right about an hour, so perfect timing. Thank you very much.
Oh, thank you for having me and allowing me to talk about cancer and what I do and everything else.
Well,
Thank you, Eileen.
we may
Thank
ask
you.
you again to come back. So thank you.
We'll do Cancer 102.
There we go. Yeah.
Good.
Thanks so much for your time, and thanks everybody else. Take care.
Okay.